So once again, the majority of output, from the ganglion cells, in your retina are derived from those P ganglion cells. Those are the parvocellular, the small ones, that are receiving, output, basically from the cone cells that are tightly packed at the foveal layer. What's also exiting as components of the optic nerves, right, are the M ganglion cell contributions, the magnocellular contributions, which are largely from peripheral sources in the retina and largely, from rod cells. Typically many of these all linked up together to a single big diffuse bipolar cell and then to one great big magnocellular M ganglion cell. And, those are also contributing a smaller number of, axons. We're talking, though, we have 4.5 to 6 million cone cells in the retina, but they contribute the bulk, of ganglion cell output, along the optic nerve. Well, there's up to 90 million rods typically. That's a lot of them, but because so many of them are all linked together, via big diffuse bipolar cells to big ganglion cells, there are fewer of those projections that are actually leaving the eye than those that are coming from cone cells in the fovea. So this again is a homuncular organization, right? We're talking again about an exaggeration of input, far more input from that foveal region that allows for high acuity color vision in daylight, right? Versus the periphery of our retina, which is much better and more active in some ways, at night, at dusk, at lower light levels, that allows us to detect motion, basically over on the side. The 90% of the projections, though, in the final ganglion cell, of course, is the intrinsically photosensitive one that's responding to blue light, right? 90% of those projections are going to the lateral geniculate nucleus in the thalamus, the little knee nucleus on the side, in the thalamus, before, the information there, from cells in the LGN, they're going to send their axons out that'll radiate. They form these they call optic radiations, big, kind of radiations of white matter that work their way back into the medial occipital lobe, along that calcerine sulcus or fissure, and synapse in layer 4, the input layer, in primary visual cortex, giving it a very thick, striped or striated appearance. That's why it's something called striate cortex, right? But 90% of humans is going that way, from the retina to the LGN to V1, right? However, there's about maybe 1% or so that is working its way, from the intrinsically photosensitive ganglion cells, right? The ones that are absorbing, largely the blue end of the spectrum, to the hypothalamus. Remember, to that suprachiasmatic nucleus, the nucleus of the hypothalamus is just above the optic chiasm, where the optic nerves are coming together and crossing, right, and becoming the optic tracts more caudally. So that's important for influencing our circadian rhythm, right? For letting us know when it's time to start drifting off to sleep and when it's time to wake up. So that projection from the intrinsically photosensitive ganglion cells out the optic nerve, about 1% of the projections in the optic nerve, to the hypothalamus, which then links up to the pineal gland and influences the release of melatonin. That's an important subcortical kind of projection, from the eyes. It's about 1% of the fibers leaving. Then there's maybe about 2% or so of the projections that are actually going from the eye to an area in the brainstem that's called the pretectal region. And this area is involved in then, sending motor output to dilate or constrict the pupil. So this is another projection, a smaller percentage of output from human eyes, right? About 2% maybe of the projections that go to this brainstem region, the pretectal region, and then that influences pupillary dilation. So it allows, sort of for automatic kind of, adjustment of the pupil so that you have the, right amount of light entering so that you can see under a large variety of, lighting conditions. And then the last kind of subcortical projection is really quite interesting. It's called the superior colliculi. Remember, the superior colliculi allow for rapid orienting, visual orienting to things in your environment. Actually, the superior colliculi are involved in what we call saccadic eye movement. So if you look around the room, you tend to jump from object to object, and that's dependent on network circuitry in the superior colliculi. But the superior colliculi are really important for kind of rapid orienting responses to novel, unexpected, threatening, surprising, kinds of visual stimuli. So if somebody throws like a ball towards your face or something, all of a sudden, you quickly, orient and deflect, something that might be coming directly towards your head. And that is actually, mediated by, about the 7% of the outputs in the retina, from the ganglion cells, back to the structure. It's on the dorsal surface of the brain, it's on the superior colliculi that mediates those fast, automated responses to visual stimuli. And it's really interesting because it turns out that people who have damage to primary visual cortex, so, that's where 90% of the output is going. If they damage primary visual cortex, they'll report that they're blind. They have no conscious visual perception. Yet, if somebody throws a ball towards their face, like a Nerf ball or something, they will actually turn and deflect. So they'll say they're blind, and yet they'll have visual reflexive responses. And I want you to kind of think about that a little bit, because much of what we do throughout the day, there's some visual conscious perceptual experience that's mediated by these cortically directed, 90% of the projections going through thalamus to, primary visual cortex, and then we're going to see further into the brain, for additional perceptual aspects. But think of how much you do visually in terms of just responding to stuff that's not conscious, that is sort of more visually reflexive behavior. I often worry about that if I'm on a bicycle and people are driving by, and they're not focused on the road, and yet they're getting enough, input to largely stay within their lane. And, a lot of this is sort of visually reflexive response, mediated by these subcortical superior colliculi directed, projections, basically, of the retina. And one last thought, humans, 90% of our projections are going into these, cortically, mediated networks that appear to be essential for sort of conscious perceptual aspects of vision, and only about 7% of these subcortical, reflexive response networks in the superior colliculi. But a cat, for example, has about 50% of the projections, coming from its eyes are actually, directed to the superior colliculi. So think of a, cat with cat toys, and, what's the benefit to having, less conscious, visual perceptual mediation of what you're observing, and more, rapid reflexive response, to visual aspects of your environment.